Hi everyone. Today we will talk some general talk about cancer in general. We will talk what is cancer. We will going to know what is the science of oncology. Also, we will deal with the meaning of how is the process that cancer may arise and how to that it is metastasized to other bodies and how the mechanism that this cancer will start and to uprise and to make its goals. This lecture will be about some biology and some histology also. We will talk about the histopathological processes of cancer at the cellular level and at the organ level. So we will start about cancer. Cancer, it is a complex disease. A complex disease, we mean by that the cancer is a strange tissue that arises abnormally in abnormal location within the body. The cancer usually starts from a single stem cell, that cell called a mutated cell, or the cell that have been attacked by carcinogens for a long time, and since carcinogens had made their goal to make mutation through this cell, and the cell change its shape, change its behavior, and change its mitotic rate. So normally every tissue in our body, there is a regulated cell growth for every tissue. In the DNA, there is some data in the genes that give the cells or that make the whole tissue, give them information that how these cells must behave to maintain the shape and the behavior and the function of the organ. Since any any toxic effect can change or alter this data, this information inside the DNA, this may arise to make cancer happen. So cancer results from the same basic process of uncontrolled growth. Cell proliferation results in a mass that invades neighboring tissues and may metastasize to more distant sites. Also, it is fatal if untreated. Cancer can make its bad effect on the tissue by two means: either by compression effect, by pressing on the surrounding organs and change or affect their function, or causes a closure of inlets and or outlets, or causes the deformity in the shape of the surrounding organs, or may by pressure effect may reduce the volume of any cavity, that like in the bladder cancer. Once the tumor arises and start to become bigger and bigger, the volume that the bladder can hold urine within it will become smaller and smaller, and so the function of the bladder will change by this way. There will be more frequent urination. So this is a change in the behavior of the organ by compression effect or by the physical mass effect. Also, we have another type of invasion of the tumor cells or the cancer cells invade through the normal tissue, and may also cause the stop or the hold of the function of that organ, or may cause abnormal function of the organ, or can cause secretions. Like for example, in some glands, the abnormal cancer or the abnormal tissue that arises within that gland will take a new function, or a different function, or an abnormal function really, and this abnormal function will cause the cancer to secrete some foreign materials or over excess of enzymes or hormones, and this will cause the new cancer will become more bad in its presence to the whole body, not only to the whole organ. Tumor formation is a multi-step process, as we said, involving many of the genetic changes in the evolving tumor cell population. Tumors are classified by site, tissue type, and degree of malignancy. Degree of malignancy, we can say by that or describe that the degree is the grade of the how much is aggressive the tumor. It could be a mild, or could be a moderate, or could be poorly differentiated, or we call it a high grade. And we have also more aggressive form is the undifferentiated type. The poorly differentiated is better than undifferentiated. Undifferentiated is more aggressive. So multi-step gene affection or mutations, the more normal cell will get increased cell growth or cell mitotic activities, and also DNA with more gene mutation, and will arise multiple cancerous effects. Even though the last step of cancer is the metastasis. The cancer cells will detach from its original primary site and go through the lymph or through bloodstream to distant organs and seed themselves there and start a new colony of cancer cells and tumor, and this colony we call it a secondary metastasis. Causes of cancer. How cancer may arise? Randomly acquired through errors in DNA replication. Many could be inherited. Could be inherited and thus present in all cells from birth. From father and mother, some cancers can be inherited, like colon cancer or breast cancer. Also have high inherited rates of happening. So, and another thing that may cause cancer, as we said, it is the carcinogen or the carcinogenic effect of some materials or environmental factors around human body, such as tobacco smoke, radiation, chemicals, or infectious agents, viruses, also are involved in cancers. So, type of mutations. The mutations in general are divided into two major groups. The germline mutations, mutation that are responsible for 5% to 10% of cancer cases. This is also called familial cancer. The germline mutations are the familial cancers. These mutations are present in every cell of the body and are passed from parent to child. The other type of the mutations in general is the sporadic cancer or the somatic mutation. So we have two types, the germline mutation, and now we have the other type, which is the somatic mutation. Somatic mutation or the sporadic cancer, which is a mutation caused by tobacco or any carcinogen, overexposure to UV radiation, ultraviolet radiation by means, and other toxins and chemicals. These are not in every cell of the body and are not passed from parent to child. These can get to every human that his environment contain these carcinogenics that causes somatic mutation. So this mutation cannot be inherited, or it is not a familial, cannot pass from parent to child. Cancer is a complex disorder, as we said, in which the normal control of cell growth is lost. Why that exactly happens, why the cell growth exactly that it changed, it is something yet not be known. But the change, the destruction in the data, in the informations, in the DNA, within the genes, it is theoretically thought to be, it is the cause that the cells will change their normal control of cell growth. Cancer is a genetic disorder, so there is a disorder in the genes, so it is a genetic disorder. Nearly all cancers are caused by abnormalities in the genetic material of the transformed cell. Initiation of cancer. Cells undergoing a series of genetic mutation or alteration which result in their instability to respond normally to intracellular, extracellular signals that control proliferation, differentiation, and death. This is the initiation of cancer. Cells undergoing a series of genetic mutation or alteration which result in their instability to respond normally to intracellular, extracellular signals that control proliferation, differentiation, and death. New aspects of the genetics of cancer pathogenesis, such as DNA methylation and microRNAs, are increasingly recognized. New genetic aspects. So, cancer develops when several genes in a cell become mutated in a way that overrides the checks and balances of the cell. Mutation inactivates tumor suppressor gene. We have something called the tumor suppressor gene. There is a gene in every cell that has a function of tumor suppression. Once this gene gets mutated, or we mean by mutated that losing its function. When that gene loses its function or become mutated, so the cells proliferate. The cells will have nothing to control its growth. The cells will proliferate like crazy, and the mutation inactivates DNA repair gene. When also there is something else, the gene that repair the DNA. Usually every cell that get damage to the DNA, it has some sort of immunity. It has some enzymes that go to make some repairs to the DNA that have been destructed. So another mutation to that gene that will repair the DNA will lose its function too. So we have two genes, the tumor suppressor gene, we have the DNA repair gene. Those two genes, when they lose their function, they will cause that thing. So mutation of a proto-oncogene creates an oncogene, also another gene. A mutation inactivates several more tumor suppressor genes. So, and the results of all these mutation or these multi-step mutations in the tumor suppressor gene or in the DNA repair gene or the proto-oncogene or inactivate several more and will cancer results. So, cancer will cause acquired functional capabilities of cancer cells, insensitivity to anti-growth signals. The body will send anti-growth signals to these cells. They will be inactive, insensitive to these signals. There is self-sufficiency in growth signals. They send to each other, the cells, the malignant cells send to each other the growth signals that are required to make the cancer grow so quickly. So there is a self-sufficiency. They reject anything that tell them from outside to stop proliferation, but they will not hear it. And the self-sufficiency, and they have their own signals, growth signals, evading apoptosis. And also they have these cells, they do not die like normal cells. The normal cells have a certain age, the certain cellular age, and then they go through the process of apoptosis. Apoptosis is the cell self-destruction. When there is any problem within it, the data that there is damage to the DNA, the cell will feel that it has the abnormal function, usually have a message to itself and make a suicide, and this we call it the apoptosis. Apoptosis will be evaded by the cancer cells. So these cells are super cells. They never die. They have their own signals. They are insensitive to anti-growth signals that come from outside. Tissue invasion and metastasis also, they grow more quickly and they can detach from the primary colony to extend through blood or through direct invasion to adjacent tissues or distant tissues. Also, they have the limitless potential for replication. The replication of the cells will be limitless, without a limit. Sustained angiogenesis. Usually when the cancer cells grow quickly and make a huge mass, this big mass will need a nutrient supply or need something that will provide it with the oxygen and other things that any cell need in the body. But once the aggregation of these cancer cells feels that there is insufficiency in providing these things around it, they start to make their own blood vessels. They give signals of the angiogenesis. Angiogenesis is the process of creating a new blood vessels for themselves from the original blood vessels of the body. So this is the neoplastic cell, this is the malignant cell, and this is how they arise and this is how they function. So there is every stem cell of cancer, the single cell that start the process of cancer will make a colony. So we talked about what is the meaning of the process of making cancer, but what we call this process. We call this process is the carcinogenesis. Carcinogenesis is what we all talked about previously. So it has 4 phases. The phase 1 is the initiation phase, where the carcinogen affect the gene, and we have the promotion phase where this malignant cell or the changed cell will pass to multiplication into proliferation, abnormal proliferation. Then the phase 3 will be a transformation phase where more genetic changes will make the cell more malignant and get more criteria of the malignancy. Then the progression phase where the true malignancy is at its full form. So as we said, the carcinogenic effect or the material or that affect the tissue, we call it toxic material, or affect the body is a toxic material, as we said. If that affect the tissue at the level tissue, we call it the carcinogenic effect. At the cellular level, we call the oncogene. Oncogene, that is the oncogene and the tumor suppressor gene and the DNA repair gene. These are the most important 3 genes within the cell that every one has function to protect the cell from being malignant cell. So an oncogene can affect the tumor suppressor gene or the DNA repair gene, as we previously talked about. So normal cell division regulated by tumor suppressor genes. The proto-oncogenes stimulate the cell growth and proliferation. Cancer, due to action of oncogenes, the proto-oncogenes get activated and oncogenes and to become oncogenes, then oncogenes increase the rate of cell growth and proliferation. Malignant, and then a malignant transformation will arise. Cancer due to action of a mutated tumor suppressor gene. Loss of or a mutation of tumor suppressor gene will cause the cell growth and proliferation to be a malignant transformation in the cell growth. So if we talk about oncogene more in detail, most oncogenes are mutated forms of normal genes called proto-oncogenes. Proto-oncogene is a normal gene that code for proteins that help to regulate cell growth and differentiation. Proto-oncogenes are often involved in signal transduction and execution of mitogenic signals. Signal transduction within the cell will be abnormal. Every connection, every messaging that will be inside the cell will be different than the normal, and this is due to the effect done by the proto-oncogenes. So proto-oncogenes can become an oncogene due to mutation or increased expression, and after activation of proto-oncogene points mutations and gene amplification and chromosomal translocation. These are all types of how the proto-oncogene will be an oncogene. Few point mutations include deletions or insertion that lead to hyperactive gene product, or deletions or insertions in the promoter region of a proto-oncogene that lead to increased transcription. By other means, any deletion or any insertion or any change in the DNA in the gene that make the gene abnormal in or cancers and its rule. So the proto-oncogene now called the oncogene. Also gene amplifications. events leading to extra chromosomal copies of a proto-oncogene lead to normal protein greatly overproduced. Amplified segments of DNA are often detected as two types of cytogenetic change: double merit and homogeneously staining regions. Also, chromosomal translocations, relocation of a proto-oncogene to a new chromosomal site that leads to a higher expression, fusion between a proto-oncogene and a second gene, which produces a fusion protein with oncogenic activity. So we understand from that fusion between a proto-oncogene and a second gene, any gene, so a proto-oncogene or nothing or a gene, this will produce a fusion protein with oncogenic activity, stronger oncogenic activity. So we have many types of oncogenes. We have the growth factors, we have growth factor receptors. These all are oncogenes. We have the GTP binding protein. We have the post-receptor tyrosine kinase. We have the cytoplasmic oncogenes. We have the nuclear oncogenes. We have the apoptotic oncogenes. These oncogenes are meant that they've lost their normal function within their origin place. So now we talk about tumor suppressor gene. Tumor suppressor gene usually, as we said, it keeps the cell safe by every change in a problem or that can cause a tumor suppression will occur. This gene is responsible for the suppression of the process of tumor formation. So normal genes implicated in the control of cell cycle repair the DNA mistakes and tell cells when to die. The product of tumor suppressor gene normally block abnormal growth and malignant transformation and lead to malignancy when the function of both alleles is lost. In general, this is a short lecture about how cancer may arise. I wish you get benefit from it, and thank you very much for your listening.